All right, so parahydrogen relayed via proton exchange and some trivia. So when I was discussing with Thomas, he said as well, that's completely okay to tell you some trivia as well about things that are going on here. And I mean, what are conferences about? Conferences are about learning new things, right? That is the scientific exchange. That is why we're here and what we learn. So I always wondered, how much do people actually understand if they see a slide, and which things they don't understand if they see a slide. So I guess the first thing, no, this doesn't, what you would not know on this slide is this. What is Kiel and where is Kiel? So that's something new to bring to you. You, of course, may know Central Europe, sort of. There we have Germany, you know Berlin, you know the Netherlands, Amsterdam, Vienna, Copenhagen, and you know there is something very special about our field, Malmö, right? That is where a lot of our things got started some 20 years ago by now. I believe that was published 2001, the first in vivo stuff. You'll see an image of that later. And there is Kiel here. So very likely none of you have heard of that before. Kiel comes from the German word Keil, which means wedge, and which is what you see here. It's the capital state of northern Germany. It's around here. It was very famous for building a lot of submarines and warships in the olden days. Now it's a beautiful city at the bay. And as there's the other big city at the bay, we are a partner town with that, with San Francisco, and we actually have a science bridge. So that's here, the science bridge between us and them. That's their Center for Intelligent Imaging. And of course, if I say San Francisco, that rings many bells with you. Hyperpolarization folks, I think, because those guys really got it clinical. And they're still going very strong. So that is Kiel, the city at the bay. That is Kiel University. It's a large city, it's a large university, 30,000 students. We are, in fact, I'm right now sitting, I'm right now sitting here behind that building. You cannot really see it, but that is where I'm sitting right now. We have some areas of what they call here excellence, or what's supposed to be the focus areas. And what's particularly interesting for us and for hyperpolarization inflammation, and of course, nanotechnology. And fortunately as well, there is Germany's second largest university hospital here. I bet you didn't know that, and I didn't know it either when I came here. It's called the UKSH, and that is, of course, very important when it comes to clinical translation. So we are the section for biomedical imaging, for clinical and preclinical imaging. We're about 30 people, headed by me and Professor Glür. That's again an image of where I'm sitting right now. My office is pretty much here. And the second one down here is the university campus from the hospital. The good thing is we have a core facility for preclinical imaging. That's the MOIN-CC, and that allows us to do animal imaging, not only with MRI, but as well as other things like ultrasound, photoacoustics, and so on. But of course, MRI and hyperpolarization is a big thing, and that it's what it should be about today. So Stefan just mentioned these small fields, and of course, they are very interesting. Here is a device that is not sold anymore. That's the Terra Nova from Macritech, which works, it detects at Earth field, and it pre-polarizes at a few millitesla. And we used that early on to do some hyperpolarized imaging of continuously rehyperpolarized Saber molecules. But that's, of course, not the end of the story. And I'm only saying this now because Stefan just brought it up. I tried once to get the gradient bore of a Bruker magnet out of the machine and just still hook it up to all the electronics of the Bruker and put like a few millitesla coil around here to get a millitesla MRI. And that sort of worked too. So I'm really interested, Stefan, to see what you get out there. This device here is quite interesting as well. That's supposed to be a teaching device from a company in southern Germany, Pure Devices. It's a 0.5 static, permanent magnet MRI. It's not so well known in our community. It's mostly for schools, but it's still very useful, I find. And of course, then there's the typical 1.5 Tesla and 3 Tesla and 7 Tesla machines here. We're very happy to having acquired a 9.4 Tesla NMR recently for our hyperpolarization experiments. And we will get an X nuclei 3 Tesla by the end of next month. So I had really a great opportunity here, or was humbled by the opportunity to build a sort of a hyperpolarization center, which will go in the ground floor of this building here, which is the ZisMed. And that is where we'll put a DMP. This one on the left side here is a picture of, I believe, the first spindle line polarizer. So that is the latest generation of DMP polarizers made by a polarized company, which is from Jan-Henrik from Copenhagen, which is a very nice device. And you actually see the polarization build up here on this window. It does it all automatically. It's very cool. And that will go there, as well as a xenon polarizer, where you see a picture in the middle, as well as our parahydrogen stuff. Here you see a mu metal device that was designed by Tim Bäumlisberger, a very capable young man, already some time ago. So that will go into this center next to the MRIs, and really interesting to see what comes out of that. Sure, these techniques are all different. So one may argue that there is not so much of synergies, but of course they are. First, it helps understanding all these different technologies, understanding one, and of course then there's the application. So these are one of our first carbon-13 imaging experiments with the 7 Tesla MRI. You see a red and you see a pyruvate and lactate image. That's still conventional CSI imaging, which is interpolated with help of post-processing from Munich. And you see the spectra below. You see the pyruvate and the lactate peak of these selected regions of interest, as well as a single voxel here. But of course, that standard CSI sequence, a sequence which I find much more intriguing, which was further developed, one has to say, by Christoph Müller, when I was still in Freiburg, is this BSSFP sequence. That goes back to the early days of hyperpolarization. I believe it was first used for chemical shift imaging in 2005 by Jochen Leupold and Jürgen Hennig. But we adapted that with 3 Tesla. And what you can see here is it's so efficient that it actually allows you to film the metabolic conversion. So what you see is we just put a phantom in the scanner, which has different vials with different amounts of lactate dehydrogenase. And then we injected pyruvate, Christoph did. And then you see this filming of this continuous imaging of the conversion. Of course, the lactate and the pyruvate, and down here you see each intensity plotted. And each of those points, it's not lines, right? Each of these points is an individual image with this high resolution acquired on the clinical scanner. And we still have quite a bit of signal after a few minutes, which I find very, very promising. Of course, our main goal is always to increase polarization to make it long-lived. But what we shall not forget is that still we need as well means to image that polarization. The conventional MRI sequences just won't do. So I'm happy to say that that is already in vivo, and hopefully we'll see some results of that published sometime soon. So parahydrogen hyperpolarization has come pretty much a long way. It all started off with the seminal work from Weitekamp. And this is just a completely subjective collection of some milestones that I found important, and which of course are leading to the point I want to make. So please don't be offended if I forgot an important paper. And it has many summers, I believe. So of course that kicked it off. That was a breakthrough. And then in the year 2001, just keep it on your tongue, right? 2001, 20 years ago, 2.0 years ago. There may be students here who weren't born then. the people from Amazon in Malmo already did in vivo imaging, and they got these beautiful angiographies. And not only that, they got as well time resolution. This is the perfusion of a pig heart, published only 5 years later. Very, very impressive very early work, I find. Then the field progressed on. We got Saber as an addition, right? First, we always were very much constrained by the needs of adding a molecule to an unsaturated bond. That was interesting, but that left the way only like for molecules like succinate from fumarate, acetyl carboxylate, and so on. But there were just not so many interesting biological agents. So that was certainly another breakthrough. And then came Silvio's work, where he introduced the sidearm approach, which was very ingenious. Stefan mentioned this as well, and this is what most of the metabolic agents today are based on. And then came another addition, which I found very intriguing. So Saber was already got rid of the need to have saturated, unsaturated precursors. And this, oh, my pen is not painting. The catalytic addition allowed to have very high polarizations. But still, Saber needed usually this nitrogen that was exchanging with the catalyst. And then Simon Duckett came up with this approach, the relay approach. So where you use, it comes in 2 flavors, in fact, where you use the reversible exchange of parahydrogen at a metal complex to polarize an intermediate, and to use that intermediate then to polarize something else. That goes with this, you basically polarize catalyst 1 and catalyst 2, or you do it via proton exchange. And that is what you see here. Sorry. Now my PowerPoint is stuck. Okay. Trying to recover. But the presentation is still ongoing. Maybe.
Yeah,
Thomas,
we're in good shape. Okay. We have enough time too, so don't get stressed.
All right, yeah. Just, maybe I need to restart PowerPoint. Discard. All right. Trying it again. Sharing my screen again. All right. Oh, I lost track of my timing, but Thomas, you will just tell me if I'm running late.
You're fine. You're totally fine. Just keep going.
Now the issue is I still cannot move the slides. No, maybe it does. So yes, we've seen this. Parahydrogen has come a long way from catalytic addition to reversible exchange to CARM13 in vivo imaging. And that was the first in vivo image, right? It was not with DMP for CARM13, it was with parahydrogen. No mention that it was acetone, the toxic agent. Then came the FIPSA, the sidearm hydrogenation, and the relay with protons. So I said that the relay comes in 2 flavors. Well, polarize complex 1, polarize catalyst 2, and polarize your target, or you polarize an agent with an exchanging hydrogen, and then you use that hydrogen to polarize your target. In this example here, glucose. Of course, glucose is it's the unachievable holy grail, if you want so. The metabolism is highly interesting. It's very important. That is what all PET imaging is based on. It's just the T1s are very short. There was one paper from Rodriguez from Kevin Brinder's group who showed it's feasible in animals. And there's very promising approaches now to use optical induced radicals for DMP to make this glucose polarization just last a little bit longer, but still it's certainly challenging. But here it was demonstrated it works as well. So I love Saber, but Saber sometimes comes with the issue that the polarization is somewhat lower than with the the covalent addition of hydrogen. The great thing of parahydrogen is its pure spin order, and if you add it to a molecule fast, you basically have it all available there, and you have more or less, you can have more or less 100% polarization. So we figured, or better, Kolja, your Kolja team, figured why not try to use the close to unity polarization of hydrogenated FIP with the broad applicability of Saber relay. So he came up with this idea of using a precursor, adding parahydrogen covalently to it, having the hyperpolarization then on the precursor, distributing it to the exchanging hydrogen, and then having a target polarized. So he, being a physicist and a chemist, he worked on magnetic particle imaging before and spin quantum simulations, but he's by education as well a chemist. He set out to work and really did an amazing job in identifying a suitable system for FIPX. So he tried 6 different transfer agents, was 3 different solvents, so really 18 combinations. And some of them had to be synthesized and so on. So that was really quite an effort. And he found out that this first system, which is propagyl alcohol and acetone D6, worked best. You'll see that here, again, the reaction scheme. You add parahydrogen to propagyl alcohol, you get the allyl alcohol, and then you have the polarization distributed within the molecule, but as well exchanging to other molecules in solution. So if you focus first on the transfer agent, here you see these peaks, and you can just guess by looking at it in the thermal spectrum. You don't see any of those. Here you see a lot of polarized peaks. He got between 10 and 40% polarization. And I'm saying here 10 or 40% because he did these experiments with 50% parahydrogen, and that will come a few more times in this session here. And he already got with this 50% parahydrogen, like 10%. Assuming that polarization would triple, which of course is an assumption, but maybe well-founded, you would get like quite a bit more. Of course, the most interesting peak is this one here, H5, the exchange in hydrogen, and that was polarized as well. You see that here, so in green, no, I should have not annotated it. It will look much better without my annotations. In green, you see the transfer agent, and this is here the polarized peak of the OH. And that was quantified already to like a few percent, so that is very promising. One important thing to keep in mind here, that this was all done just as a static magnetic field, so the transfer was just by spontaneous evolution, time evolution of the spin system, not with a sophisticated sequence as esoteric as Stefan showed in the previous presentation. So that was already sufficient to see polarized signal of ethanol and water. But to understand it more, that we found very promising. Still, we wanted to understand this, and Kolja and Andrei Pravdevtsev started to simulating these this reaction and the order transfer. So what we simulated was, in fact, we add hydrogen here, and then we observe the polarization of the proton here as a function of time, just without exchange now. And that is what you see on the center. And this is quite promising, and that even leaves us really room for experimental optimization. You see the strongest polarization was observed for 80 millitesla, and that we don't have in our setup yet in our electromagnets. So that's something where we will be going, and there we have like approaching really a few percent of polarization. We added then as well the exchange. So we had this spin system, what you see here, fully simulated with all hydrogens. And then we just assumed, of course, it doesn't stay on there. The exchange in hydrogen, it exchanges, leaves the spin system. So we simulated that with an exponential dissociation. And we found this graph here. We, and as well as some reviewers, thought isn't there something going on at very low magnetic fields? We didn't find any spontaneous transfer there. So really, it looks like we have the best transfer at moderately high fields, I would even say. So that is, I find, not so unpromising because we have already a few percent polarization for 50% parahydrogen only. So hopefully that may improve a lot if we have 100% polarization, 100% parahydrogen enrichment. So then we set out to try this. Will this work as well to polarize? Of course, we're interested in X nuclei. Protons are fine, but if we want to go for any in vivo application, we need long times. And we just took the glucose for the reason I mentioned before. It's a great molecule. It's super interesting biologically. It's very difficult for hyperpolarization, and it was done with Saber relay before. Here is again the polarization system that we used, and the glucose with the labels. And indeed, yes, we saw quite some carbon-13 polarization on the glucose, which again was very promising, I find. The polarization was approaching 1% on the carbon, so that certainly has to be improved, but we're already working with very high concentrations. So this was, we always work with about 50 millimolars. All right, so FIPX is broadly applicable. That word is missing here. That is good. We're still trying to combine these advantages that I mentioned before, the very high polarization of the covalent addition with the broad applicability. We have already quite a bit of polarization on the transfer agent. We certainly need to improve it on the target molecule, but it works as well for carbon-13. And that is all done with only 50% parahydrogen and without a dedicated transfer method. So only with the spontaneous evolution in time at the given magnetic field. All right, that is again the science part. Now I show you some more trivia. That was the arrival and the unpacking of the new DMP setup. You may recognize some hidden faces. He is very well known in the parahydrogen field. There's Andre here pushing the magnet into our building. That is a very happy Ariana Ferrari who is working on the machine. And yeah, thank you so much for your attention.
Thank you so much, Jan. Very nice presentation, very impressive work.
If there's, what is the time?
Sorry, there's still a little bit of time, so we have 5 minutes for questions and answers right now. Right. But also, there's not so many questions. I'm just going to relay this one question, and then we can keep talking. Why did you call it FIPX and not FIP relay? Yeah, of course, we had it first as FIP relay as well, but then we didn't want to impose on that message too much, right? FIP can be seen as sort of everything. Yeah, so we just said then exchange may be better. I'm open for anything. Right. So X relating to exchange and not to X nuclei. Yes, for the exchange, sorry. Yeah. Okay, good. So yeah, we do have more time. So if there's more things you want to share. Yes. Is my screen still shared? Yes. All right. So Andre came up with a very interesting approach to improve the polarization with the simple Pasadena experiment, which fortunately just got accepted. So I'm happy to share that. And that so the normal Pasadena experiment you're well aware of uses a 45-degree excitation. But if you replace that one, and that is, I'm bringing it up because I think it's good to discuss here. It's just the right audience. If you replace it with a selective 90 just on one of the hydrogens, then you can really double the polarization that you receive on either X nuclei or hydrogens. All right. So the interesting part is you excite only one. So this spectrum here, that's the normal Pasadena spectrum where you see both antiphase patterns as expected. And if you excite only one, you get only one, but higher polarized.
And you can use that then subsequently as well to improve the carbon-13 polarization.
Yeah, love it. That is very clever. That's fun, right? Like usually for Pasadena, I guess if you do a 90, you see very little signal, right? Like if you do a hard 90.
Nothing, yes, pretty much.
Right. But what you're saying is if you do a selective 90, that then you get sort of much larger signal on one of the sites, concentrated. That's a lot of fun. How did you come up with that awesome idea?
Now, that is a good question, which I don't know, I have to say. It just developed, maybe.
Very cool. Well, Andre always has, very clever ideas, so it makes sense that he would come up with this method. Fantastic. Are there any other questions?
Thomas, there's a question in the chat from Boyd.
Oh, okay. I see it here. What are the ranges of T1s for different sites and conditions of glucose?
So this is not my work. That is the work which I remember now from the Rodriguez paper, I believe in either True Nature or some Nature derivative journal. And I think the carbon-13 was just 1 or 2 seconds if it's the hydrogen, if it's the protonated molecule, and it went up to almost like 10 seconds, but not quite for the fully deuterated molecule. But please look it up. That is just based on my memory. Okay.
Well, thank you so much, Jan. This was a wonderful presentation. It's amazing that you have all these technologies in your lab, hyperpolarized xenon, hyperpolarized DNP, parahydrogen. I can't wait for all the good things to come out.
Fingers crossed. Many thanks, Thomas.